Hello, listeners. Welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and Allogene Therapeutics EVP of R&D and Chief Medical Officer, Dr. Zachary Roberts, is joining me. Zach, thanks so much for your time today. I'm so happy you're here.
Thank you, Erin. It's a pleasure.
Good. All right. Well, we're going to jump right into it. So back in June, you wrote a really terrific article for Cell and Gene that's titled "The Path Forward for CLL is Allogeneic." I highly recommend that listeners visit Cell and Gene to read that article or revisit it. Really, really well done. And in that article, you thoroughly explain that chronic lymphocytic leukemia, or CLL, is the most common leukemia in the US, and that while CLL remains a disease that is managed, it is not one that is often cured. And though patients are living longer than they once did, treatment usually eventually fails patients and, leaves a significant and growing unmet need in their relapsed refractory patient population. And so knowing all of that and, building upon what you so thoughtfully wrote in your article, I wanted to hear from you. First, let's start baseline with what are the existing issues with CAR T approaches as we are where we are today?
Yeah. So CAR T, I think it probably goes back to the beginning. The original approvals for CAR T was in 2017. These were autologous therapies, and what autologous means is that the patient themselves provide the cells, or in the lingo of the business, the starting material for manufacturing of those products. So if you are a patient who has cancer, in this case CLL, but the autologous products are also approved for certain types of non-Hodgkin's lymphoma, like diffuse large B-cell lymphoma or follicular lymphoma or mantle cell lymphoma, the patient who qualifies for this therapy will undergo a procedure called leukapheresis. Sometimes it's called apheresis. And that's a procedure where the patient sits in a chair for about 4 hours or so, and large numbers of immune cells are collected from their bloodstream. That product, the leukapheresis product, then is shipped to the manufacturer, which can be hundreds or thousands of miles away. And then when it's received there, the product is made, and that usually takes anywhere between 2 to 4 weeks in most cases, depending on the product and depending on the situation. And at the end of that process, that final CAR T product is frozen, and then it's shipped back to the treatment center where the patient originally underwent their leukapheresis. It is thawed, and then it is administered to the patient. So this was the original approach to autologous therapy. It's very much akin to autologous bone marrow transplants, and I have to say that the data for that for those products is quite astonishing. It's revolutionary. I mean, the patients who, in the case of non-Hodgkin's lymphoma, had about a 90% chance of dying from their tumors are now seeing response rates 50, 60, 70% of patients are having their tumors get better or go away altogether, and long-term survival is markedly improved. So if you fast forward now to 2024, what we're starting to see is some strains in the system. In fact, we've been seeing strains in the system for some time, and that relates to the fact that this is a bespoke manufacturing process. Every patient has to have their own product made for them. One patient, one process, one product. And that has put an enormous stress on the manufacturing infrastructure and the ability to really scale this to a degree where patients everywhere who would be eligible for this therapy have true access to it. In fact, we know that that's not the case. As many as 80+% of patients who might qualify for an autologous CAR T therapy are simply unable to get it because they don't live in the right city or they're not cared for by the right doctor. So clearly there is a need for a new therapy or a new approach. And so several years ago, Allogene and others began to develop allogeneic cell therapies. And this is at at the end, it's the same idea. You're generating a CAR T product. The main difference is that you are generating that product based on a donation from a healthy donor. So that a healthy donor comes in and undergoes that leukapheresis product. That becomes the cell starting material for manufacturing. The products are manufactured at about, in our case, 100 or more doses per healthy donation. And then those products are frozen and stored away. So the moment a patient who has a disease like relapse refractory CLL or non-Hodgkin's lymphoma is determined to need CAR T therapy, we have a product that is frozen and ready to ship. And often we can ship that product within a matter of days from the time that the patient is determined to need it. So those are that's the landscape. That's how autologous got started. And now, the need has arisen for us to move into a new technology platform to provide that access.
Good, great. And thank you for that really detailed explanation. And I want to talk a little bit more about Allo specifically. And on Cell and Gene the podcast, and actually even on Cell and Gene Live, which is our digital events that we host for Cell and Gene audience and registrants. We talk often about auto versus allo and in various ways. So for the purpose of this conversation, though, why is allogeneic the path forward, in addition to all of the, manufacturing boons and everything else? Talk to us about the path forward for Allo and why.
Absolutely. So I hinted at a couple of the benefits in the response to the last question, but I'll sort of expand on those a little bit. So starting first with access, and scalability. So as I mentioned, at Allogene, we're able to make 100 or more doses from a single manufacturing run. And in fact, as a healthy donor who in many ways has advantages over a patient with cancer because their cells have not been exposed to cycle after cycle of chemotherapy. They are not sort of suffering with the known immunosuppressive nature of metastatic cancer. So their cells are healthier, they're more abundant, and they're just a better substrate for manufacturing. So we can actually, from a single manufacturing run, we get 100 doses, but we can probably make 3 or even 4 manufacturing runs from a single healthy donor donation. So automatically, in terms of our ability to make these CAR T products for administration to patients with cancer, it's several hundred-fold more scalable. The next sort of benefit, and this is I touched on this specifically in the article that I wrote for the CLL story, was that because these cells are made from healthy donors, they themselves are freed from the long-term effect of the immunosuppressive CLL cancer. So they come into the patient more like a regular T cell. They are likely to be more activatable, if you will. They make more cytokines. They expand better than cells made from patients who have been on treatment for years and years and years, and at the time of administration have a large amount of disease. The other fact of the allogeneic product that just it makes it very attractive is that it allows us to sort of think even more broadly about the types of indications that we can potentially pursue, in large part because we can manufacture and stockpile these products. So as the field excitingly is moving in new directions, I feel like every month or certainly every few months, we're adding another indication that is treatable with CAR T. And then very recently, we've expanded into non-oncology settings with autoimmune indications suddenly attracting a lot of attention. And there, the problem gets even worse, that we're talking about not a few thousand or tens of thousands of patients. We're talking about hundreds of thousands of patients in the U.S. alone who could potentially benefit from CAR T therapy. It is really hard to imagine we as a community meeting that demand in a bespoke manufacturing platform where every patient has to have a product manufactured. The cost of goods is high. The physical structures that are required to generate these products is also very high. The last point that I'll make is that the off-the-shelf nature of an allogeneic product, and it's obviously not actually off an actual shelf, but it is accessible on demand, means that we can go into treatment centers that haven't had the wherewithal or the desire to invest in the infrastructure that's needed to offer autologous products to their patient population. So I alluded to this idea of the leukapheresis procedure, and that is a long procedure. It requires specialized equipment and trained individuals to run those units. We don't need that with an allogeneic. So many community treatment centers, what be they for oncology or for autoimmune indications, they don't have bone marrow transplant programs. They don't have leukapheresis capacity. And so for those patients and those treating physicians, they have to refer out. They have to go get treatment elsewhere. And that's actually a huge barrier for patients. So we're actually able to get into the community with these products and suddenly kind of offer a new treatment option to both patient and physician, which to date have been on the sidelines for the last almost 10 years as these products have been developed. So as we are sort of transitioning into new oncology indications and beyond, in order for us to reach those patients, we simply must have an allogeneic off-the-shelf option to meet that demand.
Yeah, sure. That makes perfect sense. And I guess piggybacking on to that part of our conversation, I want to talk about scalability specifically. In broadly, in what ways do you see allogeneic therapies delivering on scalability? And then beyond that, in Allogene's case, how is scalability feasible?
Yes. So let's again sort of compare auto versus allo, because I think it's helpful to kind of understand the core themes. So since the first autologous CAR T product was approved in 2017, that the first indication incidentally was for pediatric acute lymphoblastic leukemia, followed there very shortly by Kite being approved for diffuse large B-cell lymphoma for Yescarta. So across CD19 positive B-cell malignancies, and then there's a class of BCMA targeting autologous products for multiple myeloma. In the year 2024, cumulatively, there's probably in the neighborhood of about 30,000 patients total who have received autologous CAR T products since in the commercial setting. Allogene and Allogeneic, speaking specifically about Allogene, we have our own manufacturing facility that currently is producing products for our portfolio of clinical trials. But at the time of a potential launch of our first product, we have currently the capacity to treat 20,000 patients per year. So automatically, we're talking about an order of magnitude greater manufacturing capacity right out of the gate. And that is under the current manufacturing build. We can expand from there. So obviously, a few things have to happen between now and us treating 20,000 patients per year. But we know from where we stand today, as we are moving through our development portfolio, and I know we're likely going to cover Alpha-3 as our lead program, that's a very large number of patients. And so you need to actually, at the time of a commercial approval, you really do need to live up to the promise of CAR T and what it means to patients and physicians. You really need to be ready to treat the total number of patients who could benefit from this. And Allogene stands alone in that regard. We will have the ability to meet that demand at the time of launch.
Excellent, excellent. The in-house manufacturing facility, I'm sure, is key. And that actually, I'm glad you brought up Alpha-3, because that's exactly what I wanted to talk about next. Phase 2 Alpha-3 trial evaluating the use of cema-cel. Talk us through that.
Sure. So this is actually, I think, a great trial to really expound upon the benefits of an allogeneic off-the-shelf therapy like cema-cel. So traditionally, up until Alpha-3, every other CAR T study, really not just the ones that led to FDA approvals, but all CAR T studies that we know of, have treated patients with relapsed or refractory cancer. And that applies to multiple myeloma, it applies to non-Hodgkin's lymphoma, ALL, even some of the early phase solid tumor programs out there. Everybody who comes into those trials has measurable disease. You can see it on a scan, you can see it on a bone marrow biopsy. What we've begun to observe in the field over the last few years is that patients who have very low disease burden when they are treated with CAR T, these are patients with measurable disease, but if they have just a scant amount of cancer, tend to do better. They tend to have better efficacy outcomes. So the rates of responses and complete responses is better than their counterparts who have a lot of cancer at the time of infusion. And they tend to stay in those remissions. So that elusive cure that we are always pursuing with CAR T occurs more frequently in patients when they are treated with low disease burden. On the safety side, it also seems that those patients have less adverse events. And one of the things about CAR T is that we know that it comes with some side effects, and the famous ones are cytokine release or syndrome, or CRS, and neurotoxicity. The rates of those syndromes tend to be significantly better in patients with low disease burden. So looking for opportunities to treat patients with low disease burden is really kind of where we started when we started thinking about Alpha-3. So Alpha-3 is a unique trial, and it's a trial of patients with diffuse large B-cell lymphoma, but it is in patients who have just recently completed frontline therapy for DLBCL, or large B-cell lymphoma, not just diffuse large B-cell lymphoma. And so these patients actually are often in remission. And then for Alpha-3, all the patients who come into the study will be in remission after their frontline therapy. So they get to the end of their 6 cycles of R-CHOP or other approved regimen. They get their end of treatment scan, and their scan shows that there's no measurable cancer. What we are bringing into this trial is also a new technology. This is a technology called phased-seq, which is a type of minimal residual disease test that is being developed by our partner in this trial, Foresight Diagnostics. And what this MRD test is it's a blood test that measures circulating tumor DNA. And it's ultra-sensitive and ultra-specific for the presence of cancer. So these patients who are going to join Alpha-3, they have their scan at the end of treatment. It's negative for cancer, but they also get this blood test, this investigational blood test called phase-seq. And if that test says that there still is some cancer in their body, we are able to detect circulating tumor DNA. We know that those patients will eventually relapse. In fact, about 90% or more of them will experience a relapse of their cancer within the next 2 years. In fact, the median time to progression is on the order of a few months. So those patients are going to need more treatment. And in the best case, those patients will receive CAR T. Now, what Alpha-3 is designed to do is actually treat those patients with cema-cel, a CAR T product, in this case, an allogeneic off-the-shelf product, at the time that we detect that MRD. So what we've done here is we've bring this efficacious product that we have seen efficacy in phase 1 studies of patients with bulky disease into patients who have the lowest measurable disease that you can imagine in cancer. So we're taking advantage of the fact that those efficacy and safety outcomes tend to be better in patients with low disease burden. We're actually pushed that to its logical maximum. So the study itself is open and enrolling now. There's been a really high level of enthusiasm and engagement in the lymphoma field. Patients are excited to join because they get access to this MRD test. And the benefit to them is if the result is negative, they can be reasonably assured that their cancer is never going to come back, which itself is a huge step forward. Because in the current modality, in the current treatment paradigm, these patients don't know that. They have to keep coming back month after month to clinic, getting scans on a regular basis, literally waiting for that disease to come back. In this case, if they're MRD negative, they can be reasonably assured that that will never happen. And if they're MRD positive, we can immediately act with a dose of CAR T cells to try to eradicate that residual disease. So really a fundamentally different way to utilize CAR T, taking advantage of its off-the-shelf nature, because you have to act very quickly on this MRD positive result before the patients relapse. And finally, coming back to something I said earlier, we're using this to really push access to CAR T into community oncology practices, which don't offer CAR T right now. So these doctors are really enthusiastic about opening this trial because it means for the first time ever, they're able to offer a CAR T product to their patient without having to make the large infrastructure investments to supply their practice to provide autologous products.
That's remarkable, remarkable. Not only for the patient, but certainly for the practitioners who are administering any of the help on their side. I mean, after that incredible explanation of what's happening with Alpha-3, it's almost not a silly question. It's a great question, but at the same time, because the future looks fantastic. What's next for Allogene?
Yeah, so Allogene is in a great position right now. I think we've got an industry-leading trial and a practice-changing trial in Alpha-3. I think that alone is super exciting for me as an oncologist. But we've also got other programs up and running currently. We've got the CLL study that we talked about before. We've got a phase 1 program in solid tumors, and we're very excited to be sharing some data later this year on that program. Already, we've demonstrated probably one of the most efficacious products in solid tumor CAR T development really ever. And again, it's off the shelf, and we're able to treat these patients with metastatic kidney cancer and generate about a 30% response rate. And then we've also got a pipeline product that are very excited about. We've been talking about it for a few months now, and this is really our sort of customized, designed CAR T product for autoimmune indications, and we discussed that ever so briefly a few minutes ago. But this is one of the newest frontiers in CAR T, and bringing the potential of long-term treatment-free remissions to patients with treatment-refractory lupus and other autoimmune disorders. This is a novel product that's targeting 2 antigens, CD19, which is a B-cell antigen, and takes part in the pathogenesis of multiple different autoimmune disorders. But Allogene is unique in that we have a product that also targets CD70 in that same product, CD19 and CD70 dual targeting CAR. And this CD70 is expressed on activated T cells, which also play a role in the disease pathogenesis and autoimmune disorders. So that product is slated for an IND in the first quarter of next year, and we hope to open the study shortly thereafter and have some proof of concept data by the end of next year. So even in the short to midterm, Allogene has a number of very exciting projects ongoing right now, spanning registrational to proof of concept studies across both oncology and autoimmune indications. So that's what's next for Allogene.
Well, that's quite a bit, and really, really promising and exciting information. You have my attention, certainly Cell and Gene's audience's attention, and we will definitely be following up with you throughout 2025 and into next year to see how the progress is actually going. So fantastic.
Wonderful.
All All right, listeners, that wraps up this episode of Cell and Gene, the podcast featuring Allogene's Dr. Zachary Roberts. Zach, thank you so much for your time. This was really informative and looking forward to seeing what Allogene has coming down the pike next.
Thank you so much, Erin. This was a pleasure.
Good. Thanks for tuning in, everyone. Subscribe and download wherever you podcast, and we'll talk to you soon.